In this section, we're going to talk about the anatomy of stems and roots. So if you take a look at this image here, this shows an example of a stem cross section in red oak. And let's back up a little bit and talk really briefly about the roles of stems. Again, these are probably pretty obvious, but the first is transport of water, nutrients, carbohydrates, hormones between the roots and the top of the tree through the xylem and phloem system. Mechanical support is another function, and stems are really interesting because evolution has had to meet both that efficient transport and strong mechanical support constraints over time. And there's some interesting ways that evolution has done that. So support is another, and then storage. Again, tree stems can also be storage organs. So I'm going to talk about the main parts of the tree stem. So the first is the outer bark. The outer bark is a corky, dead cell layer that's a protective layer. It's roughly equivalent to the epidermis of the stem. And that outer bark is produced by a tissue called the cork cambium. Now, the cork cambium is not showed on this image, but the cork cambium is a meristem, and we'll talk about meristems in another lecture, but it's a tissue that produces that spongy outer bark layer. So next in from the cork cambium is the inner bark. So the inner bark is not true bark. It is not produced by the cork cambium. The inner bark is actually dead, squashed phloem tissue. So the next layer in from the inner bark is the phloem. So the inner bark is just old phloem tissue that has died, is no longer functional, and is being pressed outward, squished against the outer bark. So that's the inner bark. It's not truly bark. So the phloem is the carbohydrate transport tissue of the tree. Moving inward, another layer is the cambium. So the cambium is a thin layer of rapidly dividing cells that produces other tissues. This is another meristematic tissue. The cambium, the vascular cambium, produces the vascular tissues. It produces phloem to the outside and xylem cells to the inside. So everything inside of the vascular cambium, by definition, is xylem. So the xylem is everything inside of the vascular cambium. We'll talk about the cell types in there in a minute. In this image, you can see that they have two distinct types of phloem, or of xylem. They've got this light-colored xylem and dark-colored xylem. This light-colored xylem is sapwood. Sapwood, by definition, is just physiologically active xylem. So water transport is occurring in the tracheids or vessel elements in the sapwood, and there also are living cells. So there are living ray cells within the sapwood xylem. Heartwood, on the other hand, is physiologically inactive. It's dead. There are no living cells in the heartwood. So there's no water transport occurring in the xylem of the heartwood, and there's no living ray cells in the heartwood xylem. Sometimes in the transition from, now because all heartwood used to be sapwood, so in the transition from sapwood to heartwood, sometimes chemicals are produced that change the characteristic of the cells and change their color, decay characteristics, sometimes even smell. And that's when we have a distinctive heartwood characteristic, that's what causes it. So as an example, we have wood that has a distinctive color that usually is just attributable to the heartwood. So black walnut's a great example. Black walnut is really valued for its dark color, beautiful dark brown color. If anyone can get me a black walnut toilet seat, please do. I'd love to have one. But that black walnut, that distinctive color, just comes from the heartwood of black walnut trees. The sapwood black walnut trees is nothing special. It's just sort of that cream color. It's the heartwood that has that special color. Similarly with eastern red cedar. Red cedar is used to make cedar chests, very aromatic, repels moths that eat your wool sweaters. That's just the heartwood that has those characteristics. The sapwood of eastern red cedar, again, has that boring sapwood color. Okay. And in addition, when we say that, for instance, the wood of a species is decay resistant, generally that decay resistance is just in the heartwood of that stem. So this is a great example. This is probably a pine stem that Jason Smith found out in the woods, and someone has sawed it off. And what you can see is that decay is active in the sapwood of that pine stem. But the heartwood, the area that had undergone transition to heartwood, it's resistant to decay. And that's because in that transition from sapwood to heartwood, those chemicals that are produced in that essentially dying process, some of them are unpalatable and in some cases maybe even poisonous to decay organisms. And it's that unpalatability or that poisonous attribute that makes them resistant to decay. So again, decay resistance is generally associated with heartwood of particular tree species. Okay, so let's talk about the different cell types and their functions in xylem. So in the xylem, one cell type is phloem. So phloem consists of sieve tube cells and auxiliary cells. And these are living cells which transport carbohydrates. I already told you that the phloem tissue is just outside of the vascular cambium in gymnosperms and in dicots. In monocots, the phloem cells are in the vascular bundles and in stems, remember. So phloem tissue is alive when it's carrying out its primary function. In contrast, tracheids these are long cells which are closed at the ends and are dead when they are carrying out their primary function of water transport. Okay, so tracheids are a water transport cell and they're closed at the ends. I'm going to show you some pictures of tracheid cells. Okay, so these long cells here are tracheids, very long. They may be 1 millimeter or 2 long. So that's long for an individual cell. And they're closed at the ends. So tracheids, in order to transport water upward in the tree stem, tracheids lay next to each other. And for water to get from one tracheid to the next, they have to flow through these pits in the surface, in the cell wall of those tracheids. So just to be explicit, water transport cells like tracheids in xylem are dead, which means there's no cell membrane, there's no cell contents, there's no cytoplasm, it's just a cell wall. So basically it's a hollowed out cell wall that acts as a tube to transport water. And in the case of tracheids, they're long, narrow, and closed at the ends, and water moves from one trachea to the next through those intertracheid pits. Okay, so going back to the notes. Those are closed at the ends. Those pits are called bordered pits. And they have a special structure that allows them to act as a valve. Okay, so I'm actually going to go back to the big image because you can get a little bit better picture of it. So this is a view, the cross section of the cell wall. So if you had a tracheid like this, which is like a tube, and we cut open and opened up the cell wall, and you looked sort of at the edge of the cell wall, this is what one of those pits would look like from the side. So this is one tracheid on this side, this is another tracheid on this side, and the pit actually has a torus margo structure in it. That torus margo structure together forms what's called a bordered pit. That torus margo structure allows that pit to have a safety valve. So if we get air bubbles in the water stream in xylem, that clogs water transport and will stop water transport. So if we get an air bubble, for instance, in this tracheid, this torus margo structure will shut against the water-filled tracheid and prevent that air bubble from spreading into that water-filled tracheid. We'll talk a lot more about that process called embolism or cavitation later on, but just suffice it to say this structure acts as a valve. This is a great micrograph of the torus margo structure. In this case, you're looking, it's as if we're within the tracheid and looking straight on into a pit. So the other tracheid is on the other side of this pit. And this is the margo structure. So these are cellulose microfibrils. The water can flow through that. And this is the torus structure, which is solid. So if you get an air bubble in a tracheid, that torus margo structure closes against the water-filled tracheid. This is another great micrograph. I don't know how they get these micrograph images, but this is a great example of a torus margo structure closed shut against a water-filled and with an air-filled tracheid here. So that's an example of how they serve as a valve. Okay, so that's tracheids. Those tracheids, long, narrow, closed at the sides. Okay, another cell type in xylem are vessel members. Vessel members are also water transport cells, but in contrast to tracheids, they're open at the end. So these are all examples of vessel members, and you can see they're all open. They may have scaliform ends, but these are all open. So vessel members are wider than tracheids and shorter. Vessel members stack together hundreds or even thousands in a row to form vessels. So that's why they're called vessel members. They're subsets of a vessel. A vessel might have thousands of vessel member cells in it. So that forms a continuous wide pipe for water transport, a vessel. Vessels themselves may be up to 1 meter long. So essentially, that provides a long, uninterrupted pipe for water transport. Okay. Important characteristic of vessel members is that they only occur in angiosperms. Vessels do not occur in gymnosperms. So vessels arose with the evolution of angiosperms. And I'll talk about that in a little more detail in a second. Okay. Fiber cells are another cell type in xylem. They're also dead cells. They're just lignified cells. They look a lot like tracheids, but they're smaller. Fiber cells can occur in both angiosperms and gymnosperms. Tracheids also can occur in both angiosperms and gymnosperms as well. Fibers form or serve a support function. Okay, so I'll give you some examples of mechanical support and fibers in some cross sections in a minute. So let's talk a little bit about the evolution of vessel members and fibers. So again, conifers are older evolutionarily than angiosperms. So in conifers or gymnosperms, tracheids have to perform both the mechanical support and the water transport function. What that means is that tracheids have to have relatively thick cell walls to provide mechanical support. And what that means is that they can't be very big, because if they were big and had really thick cell walls, a lot of carbon would have to be invested in those cell walls. So those tracheids are constrained to be pretty small to be both strong and to transport water. The evolution of angiosperms, one of the innovations that evolved was the separation of the mechanical support function from the water transport function in two different cell types. So in angiosperms, the vessel members evolved and they perform water transport, while fibers perform mechanical support function. And the separation of those functions released the mechanical support constraint and allowed vessels to get really large, because they can have relatively thin and weak cell walls, because mechanical support is being provided by fibers. So that basically means that water transport cells in angiosperms are much more efficient than the water transport tissues in gymnosperms. Okay. So I wanted to talk about rays as well. So rays are another tissue type in xylem. And rays are living cells within xylem. As their name implies, they're arranged radially in files in the tree stem. They can transport carbohydrates, for instance, from storage out to the vascular cambium or inward towards like epithelial cells within the xylem. They also importantly can perform storage for especially carbohydrates within the stem. So it's those living cells within stem, living cells in the ray cells that can serve a storage function. So rays are arranged radially. So I'll give you some examples, some schematics, and some pictures of stems to sort of show you some of these structures. There's some diagrams of gymnosperm and angiosperm wood in your handouts. I'm going to show some micrographs that also really clearly show some of the structure of xylem. This is gymnosperms xylem. This is red pine. So this is the cross section on this surface. And then this is a section through the side. So if we had a cylindrical tree stem, the cross section would be looking at the end, like a cross section of that stem. But if we cut this way and looked at it from the side, that would be this. This is a radial surface if we took like a pie wedge out of it. So again, this is a gymnosperm. These are all tracheids. These are the water transport tissues of the tree. These are tracheids in cross section. And here you can see where we've exposed the side of a tracheid. You can see all those bordered pits in the side, okay? You can see a little less clearly the rays in this gymnosperm wood. This is the side of a ray right here. Those rays run radially. They run in and out. So those cells are living. All of these tracheids are dead. This is a resin canal right here. I didn't mention resin canals. I mentioned them when I talked about leaves. You can also have resin canals in gymnosperm wood as well. Okay, this is an example of angiosperm wood. Okay, this is a cross section here. These big holes in that cross section are vessel elements. So again, much wider in diameter than the tracheids in the gymnosperm wood that we saw. Much more efficient water transport. Everything, all of these cells in between those big vessels are fibers. So all of these fiber cells, some of these may be tracheids, but most of them are fibers. They're providing the mechanical support for this angiosperm wood. And the vessel elements are providing water transport, just like big pipes. You can see here's a big vessel that got cut open. Here's another vessel that got cut open. Okay. This structure here, this is a file of ray cells in cross section. Here, in this cut, you can see this is the ray cell file from a different angle. I sometimes think of files of ray cells sort of like my hand, where if I was to plunge my hand into the outside of a tree stem radially, that would be what the file of ray cells look like. So this section here would be if I like cut off the ends of my fingers and I was seeing the ends of my fingers in that file of ray cells, whereas viewing that in cross section would be like if I was viewing the edge of my hand as the file of ray cells. Okay, so that's what those cells look like. It's sometimes difficult for me to convey the three-dimensional structure and configuration of these cells. And I think some of these three-dimensional micrographs can sometimes help you envision how these cells are relative to each other. This is another example of an angiosperm xylem with big vessels, and these are a little bit smaller vessels. We'll talk about different xylem structures in a subsequent lecture. Okay, so let's talk a little bit about root structure and function. So roots, again, root functions are really not going to surprise you much. They provide anchorage for the tree, and anchorage is primarily provided by secondary or coarse roots. And that would be roots which have undergone secondary growth, or growth in width or diameter. So they're strong and provide anchorage for the tree. Obviously, the roots provide absorption of water and minerals from the soil, and they can also store carbohydrates. and nutrients. I'm not going to worry about bullet point number 4. So those are the 3 main functions of roots. And you can have different types of roots in root segments or root systems. You can have tap roots, lateral roots that come off of the tap roots. You can have sinker roots. So if you look at diagrams of root systems, you'll see those different root structures. I want to talk a little bit about aerial roots because they're pretty interesting. Aerial roots are roots that occur above ground. So in this case, these are examples of mangrove aerial roots. This is a mangrove tree, and these are mangrove aerial roots growing above the sandy beach surface here. And in this case, those mangrove aerial roots are serving as pneumatophores. Pneumatophores are just aerial roots that enable the transport of oxygen into the root system from the atmosphere. So in some tree species like mangroves, where the root systems are routinely underwater for long periods of time, the living cells in those root systems require oxygen. And there's less oxygen dissolved in the water than in the atmosphere. So the formation of aerial roots allows the transport of oxygen down into the root system so that they can be oxygenated. So in some species that have inundated root systems, you'll see the formation of aerial roots or pneumatophores. Now, interestingly, and they'll have specialized structures within them that allow the transport. There are large pore spaces in those pneumatophores that allow the diffusion of oxygen down into those root systems. Interestingly, aerial roots that are formed in bald cypress, which is another species that forms aerial roots. This is a picture of bald cypress aerial roots. These are called knees. It has not been shown that bald cypress leaves serve as pneumatophores. They don't have large air spaces in them, and they haven't been shown to really transport oxygen into bald cypress root systems. So actually the function of bald cypress knees are not known, but we don't think they're pneumatophores. And there's some argument in the literature about bald cypress knee function. Here's another example of aerial roots. This is a fig down in South Florida. Many people in my classes have seen this tree, but you can see all of these roots growing down. And this is more common in tropical, subtropical trees that maybe grow, again, in moist soils. And these aerial roots provide mechanical support for the tree, help keep it from tipping over in wet soil where anchorage from normal roots may not be as strong. So that's another interesting example of aerial roots. This is another example of aerial roots. In this case, someone leaned a motorcycle up against a fig tree or something, and that tree actually grew aerial roots around the motorcycle and then into the ground. So sort of an extreme example of aerial roots there. We'll talk really briefly about coarse roots and for fine roots. So we'll go back to the notes. So fine roots are just roots that have not undergone secondary growth. Most nutrient and water uptake occurs through fine roots. You can sort of think about the fine root system as the nutrient and water uptake part of the system, and it tends to be on the periphery of the root system, while the larger coarse roots are providing basically mechanical support and then also transport function from the fine roots out on the ends up into the stem. So interestingly, those fine roots are often very short-lived, maybe days, weeks. So they are a high turnover tissue. So they're very expensive for the tree. They are produced, function for some period of time, and then die. They're difficult to study because they're so small. So estimates of turnover time can be really difficult, but the research that's there show that they are relatively short-lived. Another thing related to root systems I want to talk about are mycorrhizae. So mycorrhizae are not technically roots. They are a symbiosis between roots and fungi. They're present on most tree species. So there is symbiosis between fungi and roots, and those fungal hyphae greatly increase the effective absorbing surface of the root system. So they basically help the root system be a more efficient absorber of water and nutrients. It may increase the specific ability to take up some nutrients, and it may protect tree roots from herbivory or disease through mechanical and potentially biochemical protection, but that's not well known. But they may form a protective function. If you think of the symbiosis here, the fungus derives carbohydrates from the roots, and the tree derives those other benefits that I've listed. So that's the, symbiotic attributes of that mycorrhizal symbiosis. Here's an example of a fine root. So you can think of this at the very end of a root system. This is a fine root that's been produced, and those fine roots can actually produce root hairs. Those root hairs are even finer. They're extensions of individual epidermal cells, and I'll show you another picture of that in a minute, but that allows them to really exploit the soil. So for instance, this is a fine root, and this is a root hair. It can actually grow between soil particles and have intimate contact with that soil matrix, which will enable it to uptake water off the surface of those soil particles and from the interstices in between them, and to take up nutrients as well. This is a good diagram showing the infection of a fine root. So this is an uninfected fine root, and this is a fine root that's been infected with a mycorrhizal hyphae. There are two main types of mycorrhizal infection. There's ectomycorrhizal and endomycorrhizal. This is an ectomycorrhizal infection, which means that the fungal hyphae do not enter the cells of the host plant. With endomycorrhizal infections, the hyphae do enter the cells, root cells. Many trees have ectomycorrhizal like this. And what you can see is that those hyphae, this is the fungal hyphae of the mycorrhizae, and they are going into the root, but between the epidermal cells of the root. So they're not actually going into the cells, they're going in between them. It's still a very intimate connection with the root, but they're not actually entering a cell. So what you can see is that that fungal ectomycorrhizal structure is well integrated with that fine root. And this is a great image that shows the extent to which mycorrhizae can expand the absorbing surface area of root systems. This is a pine germinate. So this is a young pine seedling. The brown parts here are the root system of the pine, and all of this white stuff are mycorrhizal hyphae. So you can see that mycorrhizal infection has greatly expanded the root volume that this seedling can exploit. This is a great example of the degree to which those mycorrhizal infections can advantage trees. So mycorrhizal symbiosis is really important for most tree species. I want to talk now at last very briefly about fine root structure, and I'll share the notes again so we can look at that image. So root structure, again, this is a cross section of a fine root. It hasn't undergone secondary growth yet. So this fine root has epidermal cells, just like leaves do. So that's the outer layer of cells. And then there's some ground tissue here that forms the cortex of the root. And then there's a layer called the endodermis. So endodermis just sort of means skin within. It's a skin within the root system. And the endodermis is a layer of cells that contain the Casparian strip. And this is a close-up of the Casparian strip, which occurs here in the endodermal cells. And it's just a waxy deposition in the cell walls, which prevent the inward movement of water through the cell walls. In other words, the Casparian strip or endodermis prevents the apo or the symplastic movement of water further. I apologize. It prevents the apoplastic movement of water further inward and forces water transport to become symplastic. So just to clarify that, I'll let you copy that down, but to clarify that, in the outer parts of the root system, water uptake can happen in two ways. Water can enter, for instance, a root hair or an epidermal cell and be transported across cell membranes from cell to cell, within cells, and all the way to the xylem in the inner part of the root. In contrast, water can also be taken up between cells and transported through cell walls between cells up into the point of the endodermis. At that point, because of the Casparian strip, water can't be transported through the cell walls anymore, and at the endodermis, that transport has to become symplastic, which means it has to go through a cell membrane into a cell to be transported further inward. So essentially that endodermis, that inner dermis, that inner skin layer, provides an additional layer of protection where any water being transported further in has to go through a cell membrane. And that essentially acts as a secondary filter in the water that's being taken up. Okay, so the endodermis with the Casparian strip provides some filtration against water that's being taken up before it reaches the stele which is the combination phloem-xylem tissue at the center of the primary root. So that's root structure and the end of the structure lecture.